And she was, or is currently the first female president of RANSCO, the Royal Australian New Zealand College of Ophthalmologists. And she's going to continue on the theme of treatment, but this time looking at Luxturna. So we'll see what's happening in that therapeutic space. Thanks, Heather.
Thank you, Paul. May I have my slides? Can you find my slides? Yeah, I uploaded them yesterday. Oh, what? Oh, this is good.
स्विच करना
That's it. Thank you, Paul, for that introduction, and thank you for the invitation to speak at your meeting. This is my disclosure. As you said, I'm speaking what you might call downstream from original genetic work, and I'm talking about implementing Luxturna from the perspective of a clinician and patients, and talking about these topics. So RPE65 gene, as we know, resides in the RPE and encodes a protein, RPE65. RPE65 is involved in the restoration of 11-cis retinol, so the vitamin A metabolism. And as we know, mutations in the RPE65 gene lead to degeneration of the photoreceptors in humans and in animal models. It's important to note that the phenotype of biallelic RPE65 mutation-associated retinopathy is variable, and that affects the implementation. So we see the phenotype, there's one occurring at congenital, which we call Leber congenital amaurosis. It can be seen in childhood, which we call SICORD. It can be seen presenting in adults as a typical RP. Some of the patients have a classical fundus appearance, but many do not. With this heterogeneity, the only way to be certain that you're dealing with RPE65 mutation-associated retinopathy is to do confirmatory genetic testing. And when you do that, you find that about 10% of the cases of Leber congenital amaurosis are due to RPE65. And in adult RP, about 2% of cases are due to RPE65 mutations. Everyone that works in RP knows that the mutation resides in the RPE, and secondarily we get dysfunction of the rods, and following that the cones, with this list of phenotypic features. In RPE65 mutation, because of the role of RPE65 protein, it's said that nyctalopia is more prominent than the other retinal dystrophies. Further to the point here that we have a lot of phenotypic variability, this is a study showing you the variability in decay of visual acuity and Goldman visual field over time. Now, we have, as we all know, we now have Retagene, Luxturna, as a licensed treatment for this condition. So the Veretogene is a treatment which is a live, non-replicating AAV that has been genetically modified to express the human RPE65 gene. There are many other competitors in the space beyond the form Veretogene, which has been approved by the FDA. So all the other groups have had slight differences in the construct, the vector, the purification, etc. So we need to think about the competitors as biosimilars rather than actually identical to Veretogene. Now, the phase 3 study of the Veretogene has it being given as intravitreal therapy with a vitrectomy, subretinal injection, and conceptually the product, the AAV, is transduced into the RPE cells. Some of those have the cDNA, which is incorporated into the cells, and then we have expression of normal human RPE65 protein, which has potential to restore the visual cycle. Before we look at the pivotal phase 3 study, we need to consider how we assess the effect of this therapy and what are the outcome measures when you consider that the patients with RPE65 retinopathy all have low vision at the start. So visual acuity, etc., standard measures aren't very helpful. Instead, the studies have to look at the more sophisticated outcome measures such as mobility testing, full field light sensitivity threshold testing, and functional MRI. So for the FDA-approved study, which led to Luxturna, the investigators actually worked hand in hand with the FDA to design this MLMT outcome, which is walking through a maze at varying light levels. So at baseline, all the subjects are unable to perform the maze at 1 lux, i.e. very dim circumstances. And the outcome is the improvement in the lowest light level in which they can navigate the maze, but not expressed in lux, expressed in a 6-point scale of improvement. So here are the results of the study here, the phase 3 study, which had 31 patients, 20 intervention and 9 control. Overall, 65% of the treated patients improved, but of course 66% had adverse ocular events. So if you look at the panel there with the lux score, and you can see improvement, it's not absolute lux as I said, improvement in their ability to navigate a maze in dim lighting, and a better improvement when both eyes are treated, they're the blue dots, and you can see the data out to 4 years. And similarly with the FST outcome, data out to 4 years, improvement, best improvement when both eyes are treated. You can see there the visual acuity outcome is a bit less convincing. Based on this published data, the FDA approved Luxturna, followed shortly thereafter by the European Medicines Agency, and the treatment is given to both eyes no less than 6 days apart by subretinal injection at a vitrectomy. Now, if we're looking at implementing, the durability of treatment is uncertain. The FDA-approved study showed data only out to 4 years. After that, unknown. We have longer data with the dogs, the canine model, so there's 5-year follow-up data for dogs. And interestingly, this study showed that at the treatment site, if there's 63% of photoreceptors present at the treatment site, there was a robust and long-lasting treatment response, whereas if the subretinal injection was given at areas with less than 63% photoreceptors, continuing degradation. I'm showing you here also some information from the other competitors of, as you will, biosimilars to Veretogene, not identical. But the study of Bainbridge there, which was looking at increased sensitivity on microperimetry, shows you there not all the patients responded at all. Some did, but those that responded had a decay by 3 years. And the lower panel there shows another competitor group, which when comparing control and treated areas, there was no effect on the ongoing retinal atrophy, though interestingly, even though the patients had ongoing retinal atrophy, a treatment response of increased retinal sensitivity was maintained. So I think all of this shows you that beyond 4 years, the result is uncertain. If we look at implementation, of this data, there are obvious barriers. So obviously this can only occur in specialised centres. No one has a maze in their office. The specialised centres need to have 12 mazes so they can randomly assign the patients. They need specialised pharmacy able to cover these very specialised genetically modified products, and registry for long-term follow-up. Then we have to identify the patients. Now, since it's ultra rare, the number of patients is very low. It's more or less like finding a needle in a haystack. It's said the phenotype has increased nyctalopia, but I'm yet to be convinced of that. And further, very few patients in the clinical setting have had genetic testing, and even those that have testing, it's not always certain that the mutation is pathogenic or biallelic, and certainly extensive genetic counselling is required. Beyond my scope here, obviously the best treatment window is in the younger individuals, but that's ethically problematic. The patients do not just stand up from the vitrectomy and walk away. They have to have extensive visual rehabilitation and support services. And the cost is very significant. In the USA, $850,000 for the two-eye treatment, plus the cost of surgery, plus all the follow-up, for example, the cataract surgery they'll require after vitrectomy. Working out the cost-benefit analysis is very difficult. Here, I've put here one of the models. It obviously critically depends on the assumptions made about the duration of treatment. And it comes out to be very expensive. As you can see there, $645,000 per QALY. And this needs to be interpreted in the context of opportunity cost. What else you could buy with this money? For example, a Harvard education and a self-driving car. Now, the patient experience data is none in RPE65 gene therapy. There's a little data elsewhere in gene therapy trials, which is where we look at motivation, understanding and perspective. We find the patients expecting disease reversal, which is not the case. They have poor understanding of the process, but with self-interest, they're happy to spend taxpayer dollars. So Luxturna, fabulously interesting treatment and a wonderful outcome of a whole lot of genetic research. Implementation problematic, watch this space. Thank you for listening to me, and please, you're welcome to World Ophthalmology Congress 2022, my hometown, Melbourne, Australia.
Thank you, Heather. Any questions for Dr. Mack? Yes, please.
I just have one comment. Very interesting talk. And I deal with inherited retinal diseases, so I deal with Leber congenital amaurosis as well in Florida. So I don't know. Talking to investigators later on, like the aftermarket analysis, how do you feel? What is the, I mean, objectively, we established that it was a treatment, but subjectively, how do the patients feel and how do the patients' parents feel, whether there was any improvement? I heard about your comment on the night vision, but the mobility as well.
Yeah, there are many unknowns there. I've not found published patient clinical data or patient experience data, and for me, that's the important area of follow-up, because we're trying to identify these patients out there in the wild, so to speak. Trying to identify them, we need to be looking at their patient experience, patient understanding, and then on the tiny minority who are treated, we really need to follow them up very closely to see that there's value for money. They might be better with a self-driving car and an MD from Harvard.
Correct. Well, I like to be optimistic, and I think one thing that has happened is with that price tag for the Leber congenital amaurosis treatment, there are more and more pharmaceutical companies that have suddenly become interested in inherited retinal diseases, and so that's a great thing that has happened.
Yeah, it's fantastic. Watch this space.
I think what this study shows is that certainly if you can identify mutations in genes, certainly monogenic diseases, then there is a potential to actually develop therapies. So I think we've seen the first wave of therapies now that are coming out, and I'm sure there'll be many, many more. But whether they all have the same price tag, I don't know. But I mean, $850,000 for a bilateral disease, well, it's a lot of money. But you have to weigh that up against, will it improve somebody's vision, sight, and retain it for X number of years? So this is a question for governments, health regulators, but I don't know whether it would be funded at that level in India, because it would use up a lot of your health budget, I would imagine.
Even in USA now, they are talking to the government about what should be the final price, like Medicaid or Medicare. It's not going to be that price.
And I mean, in the UK, the NIBS has done a special deal on this as well with treatment to try and lower the price. So very interesting how the dynamics work. But obviously the company needs to get its product to the market, but it has to be at a fair price, which the government can afford or stand.
Interesting comments. I think the cost-effective studies should be part of, and they are increasingly becoming. Health economics is becoming a big issue. Outcomes research has definitely been part of our fabric in the U.S. at least, and it's becoming in India as well. And see, this is where I always say that with the Indo-US collaboration, we can learn from each other. There are a lot of very cost-effective treatment, and especially with mobile devices, things are happening here at LV Prasad and Shankar Nethralaya and many places. So it's not only that the India learns from the U.S. or Australia and other countries, but we also learn from India and countries in this region. But I want to thank all the speakers. Dr. Raj Rameshwar from South Africa, he could not be here, unfortunately. So one of the things that what we do, Dr. Takeshi Iwata, who is current president of Global Eye Genetics Consortium, he always reminds me in the morning before that, please make sure that the entire audience as well as the people sitting at the podium can come in front and we get a group picture. So please join me here for the group picture. All of you, everyone, just come and please line up here. And I'm going to give my camera, and official photographer will also help us. And that will basically conclude this session. Some of us are going to be in the background over there, so please bring your questions. We are very willing, and especially Dr. Paul Baird, who's the current vice president, and he'll tell you all about GGC. So please come in front, all of you. I think you can stand up here. Yeah, please come. Yeah.
Okay, everybody, quickly do this to quickly go.
Please come, please.
Oh, yes. Nice to meet you. Yeah, yeah.
Please come. Dr. Chokshi, please come. Paul, please come in the middle. You're the official office bearer. Please come in. Please join.
Oh, quick.
Come on now.